Bioluminescence is the phenomenon of light emission by living organisms. This is well exhibited in many insects, and best understood in fireflies. Bioluminescence is the result of chemical reactions primarily involving luciferin, luciferase and oxygen. Luciferin is a heat-resistant substrate and the source of light. Luciferase, an enzyme, is the trigger, and oxygen is the fuel. Luminescing insects utilize light as a mating signal, to attract their prey, or to defend themselves from enemies. This biological phenomenon has been exploited in space and medical research, insect pest management, and is also a useful tool in biotechnology. Bioluminescence is the ability of certain animals to produce light, a phenomenon primarily seen in marine organisms. It is the predominant source of light in deep oceans. The light production is the result of chemical reactions and hence it is also called chemiluminescence. Bioluminescence is exhibited by bacteria, fungi, jellyfish, insects, algae, fish, clams, snails, crustaceans, etc. Bioluminescent bacteria have been found in marine, coastal and terrestrial environments. Some fungi can also emit light. Luminescent fungi such as Armillaria mellea and Messina spp. Produce a continuous, non-pulsing, light in their fruiting bodies and mycelium. It is believed that bioluminescent fungi use their light to attract insects that will spread the fungal spores, thus enhancing their reproduction. Some nematodes are luminescent due to the presence of symbiotic bacteria associated with them. Nematodes of the genus Neoaplectana, Steinerma and Heterorhabditis have a symbiotic association with luminescent bacteria like Xenorhabdis luminescens and thus exhibit luminescence. There are no luminous flowering plants, birds, reptiles, amphibians or mammals in nature. Though bioluminescence is generated by various organisms, it is highly RSO, NANCE, ISE. There are no luminous flowering plants, birds, reptiles, amphibians, or mammals in nature. Though bioluminescence is generated by various organisms, it is highly developed in insects. Developed in insects. The examples of true or self-luminescence are found in Collembola, Diptera, Coleoptera and Homoptera. The order Coleoptera constitutes the largest bioluminescent group in which several hundred species are known to contain highly developed photogenic organs. The best understood luminous insects belong to the families Lampyridae, Elateridae and Phengodidae. The members of Lampyridae are called fireflies or lightning bugs. Their immature forms are commonly referred to as glowworms, while the adults are called fireflies. Similarly, the individuals of Elateridae are called wireworms or click beetles. Scientists have discovered that the brightest insect is the very large Pyrophorus noctilucus, Elateridae, with a brightness of 45 millilamberts. This insect is also known as the Jamaican click beetle and the cuckooho, beetle of the West Indies. The immature forms of Phengodidae are called railroad worms. Organs of light production in insects. Bioluminescence observed in many organisms is mostly attributed to their association with luminous bacteria, but insects have evolved special photogenic organs that produce light. These organs may occur in both sexes, or may be restricted to females and immature forms, particularly in fireflies. The light organs in insects are situated very close to the body surface behind a window of translucent cuticle. They may be found scattered anywhere from the head to the tip of abdomen, including the thorax. In adult insects, these organs are mostly on the ventral side of the thorax or abdomen. In Photorus Lampyridae, males have two pairs of the light organ while females possess only one pair. Larvae and adult females of railroad worm have 11 pairs of light organs on the lateral sides of the thorax and abdomen and another pair on the head. Figure 1. In Fulgora, Homoptera, the light organ is situated only on the head. The light organs generally originate from fat bodies, except in Arachnocampa, Diptera, where these stem from the enlarged distal ends of Malpighian tubules. Approximately 50 Resonance I September 2000. Photogenic organs consist of a large number of specialized cells called follicyles, arranged cylindrically at right angles to the translucent cuticle, which permits light to pass through it. Figure 2. Behind the photocytes there is a reflecting surface chiefly consisting of urate granules. The photocytes receive oxygen through air tubes or tracheoles. They also contain a heavy aggregation of mitochondria that supplies adenosine triphosphate, ATP, required for the chemical reaction. Biochemistry of light production. Thomas Bartholin, a Danish physicist, first authored a book on animal lights as early as 1647. However, it was only in 1885 that urate granules, photocyte, trachea, mitochondria, photocyte granule epidermis, less than greater than, translucent cuticle, less than, tilde. Figure 1. The railroad worm Phrixothrix hiatus produces red light from its head and green light along the rest of its body. Photo. Copyright 1998 V. Viviani. Figure 2. A simplified cross-section of the light organ in insects. RSONA, NCE-. Thomas Bartholin, a Danish physicist, first authored a book on animal lights, as early as 1647. However, it was only in 1885 that Raphael Dubois, a French physiologist, demonstrated that three substances are involved in bioluminescence: luciferin, luciferase and molecular oxygen. Figure 3. Sequence of events involved in light production in insects. Raphael Dubois, a French physiologist, demonstrated that three substances are involved in bioluminescence: L-luciferin, luciferase and molecular oxygen. Bioluminescence is due to the substrate enzyme complex of luciferin-luciferase within the cellular cytoplasm. Luciferin refers to any light emitting -O_. Lucifer means light bringing, in Latin. These Lu-iferin Lu-iferase complexes differ in structure among species. Luciferin and luciferase of the American firefly, Photinus pyralis, was isolated in pure crystalline form by McElroy and Seliger at Johns Hopkins Institute in the early 1960s. Luciferin, which is secreted by photocytes, is a low molecular weight compound that may be an aldehyde, a polypeptide complex or a protein. Light is produced by the oxidation of luciferin in the presence of the enzyme luciferase, molecular weight 62 kDa, figure 3. ATP first activates luciferin in the presence of magnesium, Mg2+, and luciferase to produce adenoluciferin, which is then oxidized to form excited oxyluciferin. During the enzymatic oxidation of luciferin, large amounts of energy, 40 to 80 kcal per mole, become available in a single step, so that the product of the reaction is left in a highly excited state. This product emits light when the excited state returns to the ground. Luciferase plus ATP plus Mg2 plus. Luciferase plus O2. Light. Approximately 52 Resonance I September 2002. State. The reaction is very efficient, some 98% of the energy involved being released is light without production of heat. A synaptic fluid near nerve endings, which are chemical intermediaries between nerve and the light organ, triggers the light production. Variation in color of light. In most insects the light produced is yellow-green as in Photinus and Lampyris coleoptera. In larval and adult female railroad worms, the light organs on the thorax and abdomen produce green to orange light, while that on the head produces red light, figure 1. Light produced by Arachnocampa is blue-green while that of Fulgora is white. Thus, the color of light emitted varies with species and the variation may be due to environmental factors or differences in the structure of luciferase. Significance of bioluminescence in insects. In simple forms of life as in bacteria, bioluminescence is thought to have no functional significance as the light produced is a byproduct of certain chemical activity within the cell. However, in many animals, light production has known functional significance. For example, in many arthropods, the light from bioluminescence is used to lure the opposite sex for mating, or it may be used to attract prey, or for defense. Mating signal. Light is known to act as a mating signal in fireflies. In certain species the bioluminescence attracts individuals of the same species to aggregate, thus indirectly improving the chances of mating. In some species of Lampyridae, the females are wingless and sedentary, light production is therefore important for them to attract the winged moths. Flash patterns in bioluminescent insects vary between species and between sexes. On a relatively cool night, some species wait 5.5 seconds then emit a single short flash. Other species may wait 1 second and then hold the flash for a full second. Some tropical species congregate in large numbers and flash in unison. Male and female fireflies of Photinus pyralis emerge at dusk. In many arthropods, the light from bioluminescence is used to lure the opposite sex for mating, or it may be used to attract prey, or for defense. Resonance I September 2002 53. In railroad worms the continuous glow of the head region when the larvae are walking suggests a possible illumination function, whereas the circumstances under which the lateral light organs are switched on suggest a defense function, emitting a single short flash at regular intervals. The flashes are usually from male fireflies seeking mates. Males outnumber the females 50 to 1. Females climb a blade of grass, flashing when males flash within 10 to 12 feet of the females. Exchange of signals is repeated 5 to 10 times until they start ting. Predation. The most unique example of light acting as a lure for prey is found in the New Zealand glowworm fly, Arachnocampa luminosa. The female fly deposits eggs on the ceiling of dark caves. Upon hatching, the larvae hang down by a sticky thread and produce light. During night, the entire cave may glow with this light, attracting other insect species. These attracted insects get entangled in the sticky threads and are preyed upon by the larvae. The caves inhabited by flies are popularly known as luminous caves and are tourist attraction spots in New Zealand. Defense. In railroad worms the continuous glow of the head region when the larvae are walking suggests a possible illumination function, whereas the circumstances under which the lateral light organs are switched on suggest a defense function. Sudden flashes can repel potential predators. The railroad worm larvae live at high densities, confined to small areas, and may use simultaneous emission to frighten potential enemies, or they may also use the light to intimate the mated females about to lay eggs about overcrowding and competition for food sources. Applications of bioluminescence. The phenomenon of bioluminescence is utilized as a valuable scientific tool in many biological experiments. Space research. Luciferin and luciferase system can be used in spacecraft sent to Mars or other planets to explore the existence of life forms. The idea is that a special electronic device would pick up soil from the alien surface and mix it with water, oxygen, luciferin and luciferase. Then if a glow were televised back to Earth, we could know that ATP, the fifth requirement for light production, occurs there. Even as little as 1 quadrille. Approximately 54 Resonance I September 2002. Lionth of a gram of ATP is enough to be sensed by the electronic detectors. The presence of ATP would suggest, in turn, the existence of some kind of life in that alien soil that is similar to lifeforms on Earth. Medical research. The presence of ATP in every living organism has been exploited in medical research too. Injection of luciferin and luciferase exhibit different reactions in a normal and cancerous cell, and can aid in detecting energy problems in human cells. This technique is now used to study heart ailments, muscular dystrophy, urological problems, etc. The presence of ATP would suggest, in turn, the existence of some kind of life in that alien soil that is similar to lifeforms on Earth. Medical research. The presence of ATP in every living organism has been exploited in medical research too. Injection of luciferin and luciferase exhibit different reactions in a normal and cancerous cell, and can aid in detecting energy problems in human cells. This technique is now used to study heart ailments, muscular dystrophy, urological problems, etc. Pest management. Bioluminescence is used as a tool for mapping organism distribution patterns. Recently, in 2001, scientists in USA have modified the genetic material of the pink bollworm, an insect pest of cotton, with green fluorescent protein GFP, derived from the jellyfish, Acora victoria. The GFP transgenic pink bollworm strain fluoresces strongly green when viewed in its larval stage. The objective of this research is twofold. The first is to develop a GFP-marked strain of pink bollworm for field performance studies and to map the distribution of the pest. It may also serve as an additional tool for field managers. Their future objective is to eventually add a temperature-sensitive lethal gene along with the GFP gene into the pink bollworm that could be used for its management. Fluorescent marker gene. A marker gene is a short sequence of DNA that acts as a label, and it is inserted along with a gene of interest into cells. Fluorescent marker genes make the transformed cells glow under light. Genetic markers enable scientists to choose only the cells that have taken up the target genes and discard others. Introduction. When a living organism produces and emits light as a result of a chemical reaction, the process is known as bioluminescence. Bio means living, in Greek, while lumen means light, in Latin. During the process, chemical energy is converted into light energy. The process is caused by an enzyme-catalyzed chemiluminescence reaction. Bioluminescence is a product of a chemical reaction in an organism. It involves a class of chemicals called luciferins, light bringers. The luciferin oxidizes in the presence of a catalytic enzyme, luciferase, to create light in an ineffective compound, oxyluciferin. Bioluminescence (BL) serves various purposes, including sexual attraction and courtship, predation and defense. Hastings and Wilson, 1976. This process is suggested to have arisen after O2 appearance on Earth at least 30 different times during evolution, as reflected by the existence of several luciferase systems leading to different patterns of light emission, i.e. color, intensity, timing, etc. Hastings, 1983. Bioluminescence has possibly evolved from early ultra-weak chemiluminescent oxidase-catalyzed reactions to become very efficient functional light-emitting processes. Quantum yield of BL of fireflies equals 0.88, Seliger, 1975, Viviani and Baccara, 1996. A primal function in O2 detoxification, coupled with International Journal of Current Microbiology and Applied evolutionary advantages offered by specific light communication, may have allowed these species to prosper. Bioluminescence origin. The Greeks and Romans were the first to report the characteristics of luminous organisms. Aristotle 384 to 322 BC described 180 marine species and was the first to recognize cold light. The Greeks also made reference to sea phosphorescence about 500 BC. Harvey, 1957. The first book devoted to bioluminescence and chemiluminescence was published in 1555 by Conrad Gessner, 1555, Carter and Kricka, 1982, Harvey, 1957. Later in 19th century, Raphael Dubois performed a significant experiment where he extracted the two key components of a bioluminescent reaction and was able to generate light. He coined the terms luciferine and the heat labile luciferase. The first luciferin was isolated in 1956. Green and McElroy, 1956a. Structure of luciferin. Luciferins, from the Latin lucifer, light bringer, are a class of light-emitting heterocyclic compounds found in organisms that cause bioluminescence. Luciferins are a class of small molecule substrates that are oxidized in the presence of the enzyme luciferase to produce oxyluciferin and energy in the form of light. It is not known just how many types of luciferins there are, but some of the better studied compounds are listed below. There are many types of luciferins, yet all share the use of reactive oxygen species to emit light. Snail, Laetioluciferin is, in terms of chemistry, E-2-methyl-4-2, 6,6-trimethyl-1-YL-1-1-butan-1-OL formate and is from the freshwater snail Laetionaritoides. Firefly luciferin is the luciferin found in many Lampyridae species. It is the substrate of luciferase responsible for the characteristic yellow light emission from fireflies. Bacterial luciferin is a type of luciferin found in bacteria, some of which live within the specialized tissues of some squid and fish. The molecule contains a reduced riboflavin phosphate, Green and McElroy, 1956b. Cholinterrazine is found in radiolarians, ctenophores, cnidarians, squid, brittle stars, copods, chaetognaths, fish, and shrimp. It is the prosthetic group in the protein aquarin responsible for the blue light emission. Dinoflagellate luciferin is a chlorophyll derivative, i.e. a tetrapyrrole, and is found in some dinoflagellates, which are often responsible for the phenomenon of nighttime glowing waves. Historically this was called phosphorescence, but is a misleading term. Luciferins, from the Latin lucifer, light bringer, are a class of light-emitting heterocyclic compounds found in organisms that cause bioluminescence. Luciferins are a class of small molecule substrates that are oxidized in the presence of the enzyme luciferase to produce oxyluciferin and energy in the form of light. It is not known just how many types of luciferins there are, but some of the better studied compounds are listed below. There are many types of luciferins, yet all share the use of reactive oxygen species to emit light. Snail, Laetioluciferin in terms of chemistry, E-2-methyl-4-2, 6,6-trimethyl-1-YL-1-butan-1-OL formate and is from the freshwater snail Laetionaritoides. Firefly luciferin is the luciferin found in many Lampyridae species. It is the substrate of luciferase responsible for the characteristic yellow light emission from fireflies. Bacterial luciferin is a type of luciferin found in bacteria, some of which live within the specialized tissues of some squid and fish. The molecule contains a reduced riboflavin phosphate, green and McElroy, 1956b. Cholinterazine is found in radiolarians, ctenophores, cnidarians, squid, brittle stars, copods, chaetognaths, fish, and shrimp. It is the prosthetic group in the protein aquarin responsible for the blue light emission. Dinoflagellate luciferin is a chlorophyll derivative, i.e. a tetrapyrrole, and is found in some dinoflagellates, which are often responsible for the phenomenon of nighttime glowing waves. Historically this was called phosphorescence, but is a misleading term. A very similar type of luciferin is found in some types of euphausiid shrimp, Shimamura and Johnson, 1975. Vargulin is found in certain ostracods and deep-sea fish, to be specific, porichthes. Like the compound cholinterazine, it is an imidazopyrazine and emits primarily blue light in the animals. Luciferin oxidation typical for fireflies, with oxygen under luciferase catalysis in the presence of Mg2+ salts and ATP. In the first phase, luciferin reacts with oxygen under ATP consumption, Mg2+ assistance and luciferase catalysis to yield a reactive, highly unstable intermediate Int1. This intermediate is decomposing in the second stage into carbon dioxide and intermediate Int2 in excited state, phase 2. Int2 undergoes de-excitation by emission of a photon with a characteristic wavelength, phase 3. In case of luciferin, the emission maximum is at 550 to 570 nm. Diversity of bioluminescence in insects. Luminescence in terrestrial arthropods is found in Myriapoda and more extensively in insects, herring, 1987. Recently, luminescence was also reported in Amazonian species of Blatodia, Zompro and Fritz, 1999. Coleoptera beetles. The beetles have the largest number and variety of luminescent species. They are found mainly in the superfamily Elatoroidea, former Canthoroidea and Elatoroidea, which includes fireflies, Lampyridae, railroad worms, Phengodidae, click beetles, Elateridae, and related families. Luminescence has also been found in a luminescent larva of Xanthillinus in the Staphylinidae, Costa et al., 1986, and larva and adults of an undetermined species. Fireflies emit green-yellow flashes characterized by duration, interval, and frequency from ventral lanterns for sexual attraction purposes. Lloyd, 1971, Seliger, 1964, Viviani, 2001. Click beetles have two dorsal prothoracic lanterns, which usually emit continuous green light, and a ventral abdominal light organ that emits continuous green-orange light when the insect is flying. Railroad worms emit the widest range of colors among luminescent beetles, including some of the most spectacular examples with larvae and females having rows of lateral lanterns along the body emitting green-orange light and, in addition, South American species with cephalic lanterns emitting from yellow-green to red light depending on the species. In the larval stage, bioluminescence assumes mainly defensive functions, but attraction of prey has also been reported in the larvae of the Brazilian Pyrrhinister midiluminans click beetle, which display the phenomenon of luminous termite mounds. Bachara, 1988. Diptera. In Diptera, luminescence is found in the best known are Arachnocampa species from New Zealand caves and the Australasian region, Meyer, 1990, whose larvae construct webs on the roof of caves. Other luminescent mystophilids are found in the genera Chaeroplatus and Orphelia, Harvey, 1952. Orphelia fultoni is another web-building species, which occurs in stream banks of the Appalachian Mountains of eastern USA, Fulton, 1941. Functions. Larvae of Arachnocampa construct webs on the roof of caves and use their continuous blue-green luminescence to attract the flying insects on which they prey. Luminescence is produced at the terminal end of Malpighi tubules, Gatenby, 1959. When a male lands on a female pupa she glows, and when she is ready to emerge, up to 3 males may be clinging to her. A mounted male has to fight off rivals that attempt to displace him. Newly emerged females use their light to attract males if none are present. Richards, 1960. Hemiptera. Planthoppers in the family Fulgoridae, Hemiptera, Fulgoroidea, commonly called lanternflies, are busy and charismatic insects. Many fulgorid species are brilliantly colored, and some produce cuticular waxes comprised mostly of keto esters. Mason et al., 1989. Bioluminescent color variation in insect. Phengodids paired green lights on each segment and red lights at the end. Arachnocampa blue-green. Fulgora white light. Photinus and Lampyris yellow-green in color, 520-650 nanometers. Phrixothrix larval and adult female, thorax and abdomen produce green to orange light, 530-590 nanometers. That on the head produces red light, 580 nanometers to over 700 nanometers. Significance of bioluminescence in insects. In simple forms of life, bioluminescence is thought to have functional significance in many arthropods. The light from bioluminescence is used to lure the opposite sex for mating, or it may be used to attract prey, or for defense. Mating signal. Light is known to act as a mating signal in fireflies. In certain species, the bioluminescence attracts individuals of the same species to aggregate, thus indirectly improving the chances of mating. In some species of Lampyridae, the females are wingless and sedentary. Light production is therefore important for them to attract the winged males. Flash patterns in bioluminescent insects vary between species and between sexes. On a relatively cool night, some species wait 5.5 seconds then emit a single short flash. Other species may wait 1 second and then hold the flash for a full second. Some tropical species congregate in large numbers and flash in unison. Male and female fireflies of Photospiralus emerge at dusk emitting a single short flash at regular intervals. The flashes are usually from male fireflies seeking mates. Males outnumber the females 50 to 1. Females climb a blade of grass, flashing when males flash within 10 to 12 feet of the females. Exchange of signals is repeated 5 to 10 times until they start mating. Predation. The most unique example of light acting as a lure for prey is found in the New Zealand glowworm fly, Arachnocampa luminosa. The female fly deposits eggs on the ceiling of dark caves. Upon hatching, the larvae hang down by a sticky thread and produce light. During night, the entire cave may glow with this light, attracting other insect species. These attracted insects get entangled in the sticky threads and are preyed upon by the larvae. The caves inhabited by flies are popularly known as luminous caves and are tourist attraction spots in New Zealand. Defense. In railroad worms, the continuous glow of the head region when the larvae are walking suggests a possible illumination function, whereas the circumstances under which the lateral light organs are switched on suggest a defense function. Sudden flashes can repel potential predators. The railroad worm larvae live at high densities, confined to small areas, and may use simultaneous emission to frighten potential enemies, or they may also use the light to intimate the mated females about to lay eggs about overcrowding and competition for food sources. Bioluminescence in pest management. Bioluminescence is used as a tool for mapping organism distribution patterns. Recently, in 2001, scientists in USA have modified the genetic material of the pink bollworm, an insect pest of cotton, with green fluorescent protein GFP, derived from the jellyfish, Acora victoria. The GFP transgenic pink bollworm strain fluoresces strongly green when viewed in its larval stage. The objective of this research is twofold. The first is to develop a GFP-marked strain of pink bollworm for field performance studies and to map the distribution of the pest. It may also serve as an additional tool for field managers. Their future objective is to eventually add a temperature-sensitive lethal gene along with the GFP gene into the pink bollworm that could be used for its management. Babu and Cannon, 2002. Will the real firefly please stand up? Bugs that get confused with fireflies, and how to tell the difference. If it's tiny, winged, and flashing in your backyard, it might be a firefly, but it might not. Other species of bugs also glow and use bioluminescence to communicate, find mates, and scare off predators. Here are a few bugs that are commonly misidentified as fireflies, and how to tell the difference. Dailolator Physodoris gurmar. Glowing click beetles. It glows, but it isn't a firefly. It's actually a type of click beetle, dark brown. It is known as the headlight elator, because its lights are on its head. They have two small bioluminescent light organs at the back of the head and one under their abdomen. Fireflies, however, have glowing abdominal segments. Like firefly larvae, their larvae are also bioluminescent. Unlike fireflies, these glowing click beetles don't flash. However, they do seem to be able to control the intensity of the light they emit. When touched by a possible predator, for example, they become brighter. There are 3 species north of Mexico and 2 species in Florida GA region. South Texas offers the best opportunity to see these insects. Worldwide, this family is well diversified in Central and South America. Larvae of this species eat both plants and invertebrate animals, including the larvae of other species of beetle. Adults eat fermenting fruit, pollen, and smaller insects. That's another way you can tell the difference between these and fireflies. Adult fireflies are rarely, if ever, seen feeding. Railroad worms. Railroad worms. Found in both North and South America, these bugs and their larvae emit both green and red light. They're the only bioluminescent insects that emit red light. The green lights are lined up along the insect's body, while the red lights are on its head. The red light comes on when the animal is jostled or threatened, possibly to warn off predators. Mechas rodenticicalis Thomson. Firefly mimicking longhorn beetle. Probably one of the single best firefly mimics. Not only does this type of longhorn beetle resemble a firefly in size, shape, and color, it even mimics the light-producing abdominal segments with striking similarity to a firefly. Notice how this beetle has 2 yellow-colored abdominal segments. Fireflies also commonly have 2 yellow-colored light-producing segments. Amazing. Copyright Mike Quinn. Buprested. Yellow-bordered flower buprested. It looks like a firefly in the daytime but is it? This beetle bears a striking similarity to a firefly but it's not one. The scientific name for this beetle is Acmeodora flavomarginata, often listed in books as the yellow-bordered flower buprested, the term buprested designating the beetle family to which it belongs, buprested. This insect is an amazing example of mimicry, where another beetle mimics the colors of a firefly as a defense not to get eaten. Fireflies contain steroid-like compounds that are poisonous to many animals and other insects. These beetles are occasionally found on flowers and are known to be pollinators. The red blotch across the beetle's rear end is very similar to such patches on the front ends of fireflies. The yellow on the edges of its wing cover also mimic fireflies' yellow coloration on their thorax. This particular beetle was found on a frostweed weed plant located in New Braunfels, Texas, fall 2015. Drillidie. Drillidie. These bugs are not bioluminescent, but other than that, many species look a lot like fireflies, so much so that they are sometimes referred to as false firefly beetles. There are many species, and some have very obvious sexual dimorphism in Salacia unicolor, an African species of Drillidie. The female is 10 times bigger than the male. Females are wingless and look more like caterpillars than the beetles they are. Soldier beetles. Soldier beetles. These are also not bioluminescent, but they also look a lot like fireflies other than that and are related to the firefly family. A variation of the species found in Europe is bright red, reminding the British of red-coated soldiers, hence the name. They are also referred to as leatherwings, because of the leather-like texture of the wing covers on some species. The following image is a great example of a soldier beetle that is mimicking a firefly. Notice how the colors on its head look very similar to the colors of a firefly head, even down to the red, yellow, and black spot in the middle. The dark color on its thorax is also very similar to many species of fireflies. Some solider beetles such as Chalognathus pennsylvanicus, Deger, also superficially resemble fireflies in body shape and look. They have a yellow coloration with black marking on each wing cover. Glowing cockroaches. Glowing cockroaches. If you've seen this cockroach and confused it with a firefly, you were very, very lucky. Unfortunately, there is no such thing as a truly bioluminescent cockroach. Despite initial reports of the cockroach's ability to glow, a group of researchers and bioluminescence experts conducted tests on live specimens to determine if the cockroach species, Lucihormetica lucae, was indeed bioluminescent. Their finding: none of the cockroaches emitted any measurable light, Merritt 2013. Their conclusions have been confirmed by several others since then, which effectively rules out the possibility that luminous cockroaches exist. So what made people think that these cockroaches could glow in the first place? It may be due to a rare and real phenomenon, not one, however, based on the cockroach's own ability to produce biological light, but on the presence of pathological bacteria, known for almost 100 years to attack insects, especially caterpillars. Pfeiffer and Stammer 1930. The back of this cockroach's carapace is decorated with 3 spots, 1 large, 2 small, that glow when exposed to light, autofluorescence. This bacteria could preferentially colonize specific areas of the cockroach like those that cannot easily be reached by the cockroach when cleaning itself. Some have suggested that the glowing spots on the back of the cockroach could be an instance of Batesian mimicry. The glowing spots could ward off predators by mimicking a type of toxic glowing click beetle, Deileator sp. that tastes bad to most predatory species. If it looks like a firefly, and if it glows, it's not necessarily a firefly. Bioluminescence is rarer on land than in the water, but there are several species of bug that emit their own light in addition to fireflies. Some do it through chemical processes that take place in their bodies, as fireflies do. Others, such as the exceedingly rare glowing cockroach, rely on bioluminescent bacteria. No matter how they do it, however, they're all beautiful in their own way, and fascinating. Fun fact. How to catch a firefly. Get prepared for this year. Fireflies start appearing as early as April and May in the U.S. Learn tips on how best to catch and release fireflies. Learn. Firefly fact. Number of species. In North America, there are an estimated 270 plus species. Worldwide there is estimated 2,400 plus species. These numbers are going up as new Lampyridae species are described by scientists. Fun fact. Why blink? The purpose of blinking is so that male fireflies can find potential mates. If you watch long enough you will be able to pick up on their unique flash pattern. Try to remember it so that next time you see a firefly flash you can identify it. Get involved. Have you seen a firefly? Firefly scientists are in urgent need of your observations. Become a citizen scientist and record your sighting using iNaturalist. Using our helpful guide you'll learn what you need. Will the real firefly please stand up? Bioluminescence. Bioluminescence is the production and emission of light by living organisms. It is a form of chemiluminescence. Bioluminescence occurs widely in marine vertebrates and invertebrates, as well as in some fungi, microorganisms including some bioluminescent bacteria, and terrestrial arthropods such as fireflies. In some animals, the light is bacteriogenic, produced by symbiotic bacteria such as those from the genus Vibrio. In others, it is autogenic, produced by the animals themselves. Flying and glowing firefly, Photinus pyrolis. Female glowworm, Lampyris noctiluca. In a general sense, the principal chemical reaction in bioluminescence involves a light-emitting molecule and an enzyme, generally called luciferin and luciferase, respectively. Because these are generic names, luciferins and luciferases are often distinguished by the species or group, e.g. firefly luciferin. In all characterized cases, the enzyme catalyzes the oxidation of the luciferin. In some species, the luciferase requires other cofactors, such as calcium or magnesium ions, and sometimes also the energy-carrying molecule adenosine triphosphate, ATP. In evolution, luciferins vary little. One in particular, cholinterazine, is found in 11 different animal phyla, though in some of these, the animals obtain it through their diet. Conversely, luciferases vary widely between different species. Bioluminescence has arisen over 40 times in evolutionary history. Both Aristotle and Pliny the Elder mentioned that damp wood sometimes gives off a glow. Many centuries later, Robert Boyle showed that oxygen was involved in the process, in both wood and glowworms. It was not until the late 19th century that bioluminescence was properly investigated. The phenomenon is widely distributed among animal groups, especially in marine environments. On land it occurs in fungi, bacteria and some groups of invertebrates, including insects. The uses of bioluminescence by animals include counterillumination camouflage, mimicry of other animals, for example to lure prey, and signaling to other individuals of the same species, such as to attract mates. In the laboratory, luciferase-based systems are used in genetic engineering and biomedical research. Researchers are also investigating the possibility of using bioluminescent systems for street and decorative lighting, and a bioluminescent plant has been created. 2. History. Before the development of the safety lamp for use in coal mines, dried fish skins were used in Britain and Europe as a weak source of light. 3. This experimental form of illumination avoided the necessity of using candles which risked sparking explosions of fire damp. 4. In 1920, the American zoologist E. Newton Harvey published a monograph, The Nature of Animal Light, summarizing early work on bioluminescence. Harvey notes that Aristotle mentions light produced by dead fish and flesh, and that both Aristotle and Pliny the Elder, in his Natural History, mention light from damp wood. He records that Robert Boyle experimented on these light sources, and showed that both they and the glowworm require air for light to be produced. Harvey notes that in 1753, J. Baker identified the flagellate Noctiluca as a luminous animal, just visible to the naked eye. 5. And in 1854 Johann Florian Heller, 1813-1871, identified strands, hyphae, of fungi as the source of light in dead wood. 6. Tuckey, in his posthumous 1818 narrative of the expedition to the Zaire, described catching the animals responsible for luminescence. He mentions pellucids, crustaceans to which he ascribes the milky whiteness of the water, and cancers, shrimps and crabs. Under the microscope he described the luminous property to be in the brain, resembling a most brilliant amethyst about the size of a large pin's head. 7. Charles Darwin noticed bioluminescence in the sea, describing it in his journal. While sailing in these latitudes on one very dark night, the sea presented a wonderful and most beautiful spectacle. There was a fresh breeze, and every part of the surface, which during the day is seen as foam, now glowed with a pale light. The vessel drove before her bows 2 billows of liquid phosphorus, and in her wake she was followed by a milky train. As far as the eye reached, the crest of every wave was bright, and the sky above the horizon, from the reflected glare of these livid flames, was not so utterly obscure, as over the rest of the heavens. 8. Darwin also observed a luminous jellyfish of the genus Dionea, 8, noting that, when the waves scintillate with bright green sparks, I believe it is generally owing to minute crustacea. But there can be no doubt that very many other pelagic animals, when alive, are phosphorescent. 8. He guessed that a disturbed electrical condition of the atmosphere, 8, was probably responsible. Daniel Pauly comments that Darwin was lucky with most of his guesses, but not here. 9. Noting that biochemistry was too little known, and that the complex evolution of the marine animals involved would have been too much for comfort. 9. Osamu Shimamura isolated the photoprotein aquarin and its cofactor cholinterazine from the crystal jelly Echeria victoria in 1961. 10. Bioluminescence attracted the attention of the United States Navy in the Cold War, since submarines in some waters can create a bright enough wake to be detected. A German submarine was sunk in the First World War, having been detected in this way. The Navy was interested in predicting when such detection would be possible, and hence guiding their own submarines to avoid detection. 11. Among the anecdotes of navigation by bioluminescence is one recounted by the Apollo 13 astronaut Jim Lovell, who as a Navy pilot had found his way back to his aircraft carrier USS Shangri-La when his navigation systems failed. Turning off his cabin lights, he saw the glowing wake of the ship, and was able to fly to it and land safely. 12. The French pharmacologist Raphael Dubois carried out work on bioluminescence in the late 19th century. He studied click beetles Pyrophorus and the marine bivalve mollusk Pholis dactylus. He refuted the old idea that bioluminescence came from phosphorus, 13, and demonstrated that the process was related to the oxidation of a specific compound, which he named luciferin, by an enzyme. 15. He sent Harvey siphons from the mollusk preserved in sugar. Harvey had become interested in bioluminescence as a result of visiting the South Pacific and Japan and observing phosphorescent organisms there. He studied the phenomenon for many years. His research aimed to demonstrate that luciferin, and the enzymes that act on it to produce light, were interchangeable between species, showing that all bioluminescent organisms had a common ancestor. However, he found this hypothesis to be false, with different organisms having major differences in the composition of their light-producing proteins. He spent the next 30 years purifying and studying the components, but it fell to the young Japanese chemist Osamu Shimamura to be the first to obtain crystalline luciferin. He used the sea firefly Vargula hilgendorfii, but it was another 10 years before he discovered the chemical structure and published his 1957 paper Crystalline Cypridina luciferin. 16. Shimamura, Martin Chalfie, and Roger Y. Chen won the 2008 Nobel Prize in Chemistry for their 1961 discovery and development of green fluorescent protein as a tool for biological research. 17. Harvey wrote a detailed historical account on all forms of luminescence in 1957. 18. An updated book on bioluminescence covering also the 20th and early 21st century was published recently. 1920. Evolution. In 1932 E. N. Harvey was among the first to propose how bioluminescence could have evolved. 21. In Thieserly paper, he suggested that protobioluminescence could have arisen from respiratory chain proteins that hold fluorescent groups. This hypothesis has since been disproven, but it did lead to considerable interest in the origins of the phenomenon. Today, the two prevailing hypotheses, both concerning marine bioluminescence, are those put forth by Howard Seliger in 1993 and Rees et al. In 1998. 22-23. Seliger's theory identifies luciferase enzymes as the catalyst for the evolution of bioluminescent systems. It suggests that the original purpose of luciferases was as mixed-function oxygenases. As the early ancestors of many species moved into deeper and darker waters, natural selection favored the development of increased eye sensitivity and enhanced visual signals. 24. If selection were to favor a mutation in the oxygenase enzyme required for the breakdown of pigment molecules, molecules often associated with spots used to attract a mate or distract a predator, it could have eventually resulted in external luminescence in tissues. 22. Rees et al. Use evidence gathered from the marine luciferin cholinterazine to suggest that selection acting in luciferins may have arisen from pressures to protect oceanic organisms from potentially deleterious reactive oxygen species, e.g. H2O2 and O2. Minus. The functional shift from antioxidation to bioluminescence probably occurred when the strength of selection for antioxidation defense decreased as early species moved further down the water column. At greater depths, exposure to ROS is significantly lower, as is the endogenous production of ROS through metabolism. 23. While popular at first, Seliger's theory has been challenged, particularly on the biochemical and genetic evidence that Rees examines. What remains clear, however, is that bioluminescence has evolved independently at least 40 times. 25. Bioluminescence in fish began at least by the Cretaceous period. About 1,500 fish species are known to be bioluminescent. The capability evolved independently at least 27 times. Of these, 17 involved the taking up of bioluminous bacteria from the surrounding water, while in the others, the intrinsic light evolved through chemical synthesis. These fish have become surprisingly diverse in the deep ocean and control their light with the help of their nervous system, using it not just to lure prey or hide from predators, but also for communication. 26-27. All bioluminescent organisms have in common that the reaction of a luciferin and oxygen is catalyzed by a luciferase to produce light. 28. McElroy and Seliger proposed in 1962 that the bioluminescent reaction evolved to detoxify oxygen, in parallel with photosynthesis. 29. Thusen, Davis et al. Showed in 2016 that bioluminescence has evolved independently 27 times within 14 fish clades across ray fin fishes. 26. The oldest of these appears to be stomiforms and myctophyty. 30. In sharks, bioluminescence has evolved only once. 31. Genomic analysis of octocorals indicates that their ancestor was bioluminescent as long as 540 million years ago. 32. Chemical mechanism. Protein structure of the luciferase of the firefly Photinus pyrolis. The enzyme is a much larger molecule than luciferin. Bioluminescence is a form of chemiluminescence where light energy is released by a chemical reaction. This reaction involves a light-emitting pigment, the luciferin, and a luciferase, the enzyme component. 33. Because of the diversity of luciferin-luciferase combinations, there are very few commonalities in the chemical mechanism. From currently studied systems, the only unifying mechanism is the role of molecular oxygen. Often there is a concurrent release of carbon dioxide, CO2. For example, the firefly luciferin-luciferase reaction requires magnesium and ATP and produces CO2, adenosine monophosphate, AMP, and pyrophosphate, PP, as waste products. Other cofactors may be required, such as calcium, Ca2+, for the photoprotein aquarin, or magnesium, Mg2+, ions and ATP for the firefly luciferase. 34. Generically, this reaction can be described as luciferin plus O2 oxyluciferin plus light energy. Cholinterazine is a luciferin found in many different marine phyla from comb jellies to vertebrates. Like all luciferins, it is oxidized to produce light. Instead of a luciferase, the jellyfish Echeria victoria makes use of another type of protein called a photoprotein, in this case specifically aquarin. 35. When calcium ions are added, rapid catalysis creates a brief flash quite unlike the prolonged glow produced by luciferase. In a second, much slower step, luciferin is regenerated from the oxidized oxyluciferin form, allowing it to recombine with aquarin, in preparation for a subsequent flash. Photoproteins are thus enzymes, but with unusual reaction kinetics. 36. Furthermore, some of the blue light released by aquarin in contact with calcium ions is absorbed by a green fluorescent protein, which in turn releases green light in a process called resonant energy transfer. 37. Overall, bioluminescence has arisen over 40 times in evolutionary history. 33. In evolution, luciferins tend to vary little. One in particular, cholinterazine, is the light-emitting pigment for 9 phyla, groups of very different organisms, including polycystine radiolaria, Cercozoa, Piodaria, Protozoa, comb jellies, Cnidaria including jellyfish and corals, crustaceans, mollusks, arrowworms and vertebrates, ray-finned fish. Not all these organisms synthesize cholinterazine. Some of them obtain it through their diet. 33. Conversely, luciferase enzymes vary widely and tend to be different in each species. 33. Distribution. Huge numbers of dinoflagellates creating bioluminescence in breaking waves. Bioluminescence occurs widely among animals, especially in the open sea, including fish, jellyfish, comb jellies, crustaceans, and cephalopod mollusks, in some fungi and bacteria, and in various terrestrial invertebrates, nearly all of which are beetles. In marine coastal habitats, about 2.5% of organisms are estimated to be bioluminescent, whereas in pelagic habitats in the eastern Pacific, about 76% of the main taxa of deep-sea animals have been found to be capable of producing light. 38. More than 700 animal genera have been recorded with light-producing species. 39. Most marine light emission is in the blue and green light spectrum. However, some loose-jawed fish emit red and infrared light, and the genus Tomopteris emits yellow light. 33. 40. The most frequently encountered bioluminescent organisms may be the dinoflagellates in the surface layers of the sea, which are responsible for the sparkling luminescence sometimes seen at night in disturbed water. At least 18 genera of these phytoplankton exhibit luminosity. 33. Luminescent dinoflagellate ecosystems are present in warm water lagoons and bays with narrow openings to the ocean. 41. A different effect is the thousands of square miles of the ocean which shine with the light produced by bioluminescent bacteria, known as Mariel or the Milky Seas effect. 42. Pelagic zone. Bioluminescence is abundant in the pelagic zone, with the most concentration at depths devoid of light and surface waters at night. These organisms participate in diurnal vertical migration from the dark depths to the surface at night, dispersing the population of bioluminescent organisms across the pelagic water column. The dispersal of bioluminescence across different depths in the pelagic zone has been attributed to the selection pressures imposed by predation and the lack of places to hide in the open sea. In depths where sunlight never penetrates, often below 200 meters, the significance of bioluminescence is evident in the retainment of functional eyes for organisms to detect bioluminescence. 43. Bacterial symbiosis. Organisms often produce bioluminescence themselves, rarely do they generate it from outside phenomena. However, there are occasions where bioluminescence is produced by bacterial symbionts that have a symbiotic relationship with the host organism. Although many luminous bacteria in the marine environment are free-living, a majority are found in symbiotic relationships that involve fish, squids, crustaceans, etc. as hosts. Most luminous bacteria inhabit the sea, dominated by Photobacterium and Vibrio. 44. In the symbiotic relationship, bacterium benefit from having a source of nourishment and a refuge to grow. Hosts obtain these bacterial symbionts either from the environment, spawning, or the luminous bacterium is evolving with their host. 45. Coevolutionary interactions are suggested as host organisms' anatomical adaptations have become specific to only certain luminous bacteria, to suffice ecological dependence of bioluminescence. 46. Benthic zone. Bioluminescence is widely studied amongst species located in the mesopelagic zone, but the benthic zone at mesopelagic depths has remained widely unknown. Benthic habitats at depths beyond the mesopelagic are also poorly understood due to the same constraints. Unlike the pelagic zone where the emission of light is undisturbed in the open sea, the occurrence of bioluminescence in the benthic zone is less common. It has been attributed to the blockage of emitted light by a number of sources such as the sea floor, and inorganic and organic structures. Visual signals and communication that is prevalent in the pelagic zone such as counter-illumination may not be functional or relevant in the benthic realm. Bioluminescence in bathyal benthic species still remains poorly studied due to difficulties of the collection of species at these depths. 47. Uses in nature. Messina chlorophyllus, a bioluminescent mushroom. Bioluminescence has several functions in different taxa. Stephen Haddock et al. 2010 list as more or less definite functions in marine organisms the following: defensive functions of startle, counter-illumination, camouflage, misdirection, smoke screen, distractive body parts, burglar alarm, making predators easier for higher predators to see, and warning to deter settlers, offensive function, soft lure, stun or confuse prey, illuminate. Bioluminescence has several functions in different taxa. Stephen Haddock et al. 2010 list as more or less definite functions in marine organisms the following: defensive functions of startle, counter-illumination, camouflage, misdirection, smoke screen, distractive body parts, burglar alarm, making predators easier for higher predators to see, and warning to deter settlers, offensive function, soft lure, stun or confuse prey, illuminate prey, and mate attraction, recognition. It is much easier for researchers to detect that a species is able to produce light than to analyze the chemical mechanisms or to prove what function the light serves. 33. In some cases the function is unknown. A Swiss species in 3 families of earthworm, Oligocata, such as Diplocardia longa, where the coalomic fluid produces light when the animal moves. 48. The following functions are reasonably well established in the named organisms. Counter-illumination, camouflage. Principle of counter-illumination camouflage in firefly squid, Wattisenia scintillans. When seen from below by a predator, the bioluminescence helps to match the squid's brightness and color to the sea surface above. In many animals of the deep sea, including several squid species, bacterial bioluminescence is used for camouflage by counter-illumination, in which the animal matches the overhead environmental light as seen from below. 49. In these animals, photoreceptors control the illumination to match the brightness of the background. 49. These light organs are usually separate from the tissue containing the bioluminescent bacteria. However, in one species, Euprymna scolopes, the bacteria are an integral component of the animal's light organ. 50. Attraction. Storoteuthis cirtensis bioluminescent photophores. Bioluminescence is used in a variety of ways and for different purposes. The serrate octopod Storoteuthis cirtensis emits bioluminescence from its sucker-like structures. 51. These structures are believed to have evolved from what are more commonly known as octopus suckers. They do not have the same function as the normal suckers because they no longer have any handling or grappling ability due its evolution of photophores. The placement of the photophores are within the animal's oral reach, which leads researchers to suggest that it uses its bioluminescence to capture and lure prey. 52. Fireflies use light to attract mates. Two systems are involved according to species. In one, females emit light from their abdomens to attract males. In the other, flying males emit signals to which the sometimes sedentary females respond. 4853. Click beetles emit an orange light from the abdomen when flying and a green light from the thorax when they are disturbed or moving about on the ground. The former is probably a sexual attractant, but the latter may be defensive. 48. Larvae of the click beetle Pyrophorus nyctiphanus live in the surface layers of termite mounds in Brazil. They light up the mounds by emitting a bright greenish glow which attracts the flying insects on which they feed. 48. In the marine environment, use of luminescence for mate attraction is chiefly known among ostracods, small shrimp-like crustaceans, especially in the family Cyprididae. Pheromones may be used for long-distance communication, with bioluminescence used at close range to enable mates to home in. 33. A polychaete worm. The Bermuda fireworm creates a brief display, a few nights after the full moon, when the female lights up to attract males. 54. Defense. Acanthophira purpurea has photophores along its body which it uses in defense against predators. The defense mechanisms for bioluminescent organisms can come in multiple forms: startling prey, counter-illumination, smoke screen or misdirection, distractive body parts, burglar alarm, sacrificial tag or warning coloration. The shrimp family Oplophoridae use their bioluminescence as a way of startling the predator that is after them. 55. Acanthophira purpurea, within the Oplophoridae family, uses its photophores to emit light, and can secrete a bioluminescent substance when in the presence of a predator. This secretory mechanism is common among prey fish. 55. Many cephalopods, including at least 70 genera of squid, are bioluminescent. 33. Some squid and small crustaceans use bioluminescent chemical mixtures or bacterial slurries in the same way as many squid use ink. A cloud of luminescent material is expelled, distracting or repelling a potential predator, while the animal escapes to safety. 33. The deep-sea squid Octopotuthis delatron may autotomize portions of its arms which are luminous and continue to twitch and flash, thus distracting a predator while the animal flees. 33. Dinoflagellates may use bioluminescence for defense against predators. They shine when they detect a predator, possibly making the predator itself more vulnerable by attracting the attention of predators from higher trophic levels. 33. Grazing copepods release any phytoplankton cells that flash, unharmed. If they were eaten they would make the copepods glow, attracting predators, so the phytoplankton's bioluminescence is defensive. The problem of shining stomach contents is solved, and the explanation corroborated. In predatory deep-sea fishes, their stomachs have a black lining able to keep the light from any bioluminescent fish prey which they have swallowed from attracting larger predators. 9. The sea firefly is a small crustacean living in sediment. At rest it emits a dull glow, but when disturbed it darts away leaving a cloud of shimmering blue light to confuse the predator. During World War II it was gathered and dried for use by the Japanese army as a source of light during clandestine operations. 16. The larvae of railroad worms, Phrixothrix, have paired photic organs on each body segment, able to glow with green light. These are thought to have a defensive purpose. 56. They also have organs on the head which produce red light. They are the only terrestrial organisms to emit light of this color. 57. Warning. Aposematism is a widely used function of bioluminescence, providing a warning that the creature concerned is unpalatable. It is suggested that many firefly larvae glow to repel predators. Some millipedes glow for the same purpose. 58. Some marine organisms are believed to emit light for a similar reason. These include scale worms, jellyfish and brittle stars, but further research is needed to fully establish the function of the luminescence. Such a mechanism would be of particular advantage to soft-bodied cnidarians if they were able to deter predation in this way. 33. The limpet Latia neretoides is the only known freshwater gastropod that emits light. It produces greenish luminescent mucus which may have an anti-predator function. 59. The marine snail Hinia brasiliana uses flashes of light, probably to deter predators. The blue-green light is emitted through the translucent shell, which functions as an efficient diffuser of light. 60. Communication. Pyrosoma, a colonial tunicate, each individual zooid in the colony flashes a blue-green light. Communication in the form of quorum sensing plays a role in the regulation of luminescence in many species of bacteria. Small extracellularly secreted molecules stimulate the bacteria to turn on genes for light production when cell density, measured by concentration of the secreted molecules, is high. 33. Pyrosomes are colonial tunicates and each zooid has a pair of luminescent organs on either side of the inlet siphon. When stimulated by light, these turn on and off, causing rhythmic flashing. No neural pathway runs between the zooids, but each responds to the light produced by other individuals, and even to light from other nearby colonies. 61. Communication by light emission between the zooids enables coordination of colony effort, for example in swimming where each zooid provides part of the propulsive force. 62. Some bioluminous bacteria infect nematodes that parasitize Lepidoptera larvae. When these caterpillars die, their luminosity may attract predators to the dead insect, thus assisting in the dispersal of both bacteria and nematodes. 48. A similar reason may account for the many species of fungi that emit light. Species in the genera Armillaria, Messina, Omphalodus, Pinellus, Pleurotus and others do this, emitting usually greenish light from the mycelium, cap and gills. This may attract night-flying insects and aid in spore dispersal, but other functions may also be involved. 48. Quantula striata is the only known bioluminescent terrestrial mollusk. Pulses of light are emitted from a gland near the front of the foot and may have a communicative function, although the adaptive significance is not fully understood. 63. Mimicry. A deep-sea anglerfish, Buphoceratias wedley, showing the esca lure. Bioluminescence is used by a variety of animals to mimic other species. Many species of deep-sea fish such as the anglerfish and dragonfish make use of aggressive mimicry to attract prey. They have an appendage on their heads called an esca that contains bioluminescent bacteria able to produce a long-lasting glow which the fish can control. The glowing esca is dangled or waved about to lure small animals to within striking distance of the fish. 33.64. The cookie-cutter shark uses bioluminescence to camouflage its underside by counter-illumination, but a small patch near its pectoral fins remains dark, appearing as a small fish to large predatory fish like tuna and mackerel swimming beneath it. When such fish approach the lure, they are bitten by the shark. 65.66. Female Photorus fireflies sometimes mimic the light pattern of another firefly, Photinus, to attract its males as prey. In this way they obtain both food and the defensive chemicals named lucibufagins, which Photorus cannot synthesize. 67. South American giant cockroaches of the genus Lucihormetica were believed to be the first known example of defensive mimicry, emitting light in imitation of bioluminescent, poisonous click beetles. 68. However, doubt has been cast on this assertion, and there is no conclusive evidence that the cockroaches are bioluminescent. 69.70. Flashing of photophores of black dragonfish, Mollucosteus niger, showing red fluorescence. Illumination. While most marine bioluminescence is green to blue, some deep-sea barbel dragonfishes in the genera Aristostomias, Pachystomias and Mollucosteus emit a red glow. This adaptation allows the fish to see red pigmented prey, which are normally invisible to other organisms in the deep ocean environment where red light has been filtered out by the water column. 71. These fish are able to utilize the longer wavelength to act as a spotlight for its prey that only they can see. 71. The fish may also use this light to communicate with each other to find potential mates. 72. The ability of the fish to see this light is explained by the presence of specialized rhodopsin pigment. 71. The mechanism of light creation is through a suborbital photophore that utilizes gland cells which produce exergonic chemical reactions that produce light with a longer, red wavelength. 73. The dragonfish species which produce the red light also produce blue light in photophore on the dorsal area. 73. The main function of this is to alert the fish to the presence of its prey. 74. The additional pigment is thought to be assimilated from chlorophyll derivatives found in the copods which form part of its diet. 74. The angler siphonophore, Arena, utilizes red bioluminescence in appendages to lure fish. 73. Biotechnology. Biology and medicine. Bioluminescent organisms are a target for many areas of research. Luciferase systems are widely used in genetic engineering as reporter genes, each producing a different color by fluorescence, 75-76, and for biomedical research using bioluminescence imaging. 77, 78, 79. For example, the firefly luciferase gene was used as early as 1986 for research using transgenic tobacco plants. 80. Vibrio bacteria symbiose with marine invertebrates such as the Hawaiian bobtail squid, Euprymnus scolopes, are key experimental models for bioluminescence. 81, 82. Bioluminescent activated destruction is an experimental cancer treatment. 83. In vivo luminescence cell and animal imaging uses dyes and fluorescent proteins as chromophores. The characteristics of each chromophore determine which cell areas will be targeted and illuminated. 84. Light production. A firefly, Petunia, genetically engineered to produce luciferase. The structures of photophores, the light-producing organs in bioluminescent organisms, are being investigated by industrial designers. Engineered bioluminescence could perhaps one day be used to reduce the need for street lighting, or for decorative purposes if it becomes possible to produce light that is both bright enough and can be sustained for long periods at a workable price. 11. 85, 86. The gene that makes the tails of fireflies glow has been added to mustard plants. The plants glow faintly for an hour when touched, but a sensitive camera is needed to see the glow. 87. University of Wisconsin-Madison is researching the use of genetically engineered bioluminescent E. coli bacteria, for use as bioluminescent bacteria in a light bulb. 88. In 2011, Philips launched a microbial system for ambience lighting in the home. 89, 90. An IGAM team from Cambridge, England, has started to address the problem that luciferin is consumed in the light-producing reaction by developing a genetic biotechnology part that codes for a luciferin-regenerating enzyme from the North American firefly. 91. In 2016, Glowy, a French company, started selling bioluminescent lights for shop fronts and street signs. 92. For use between 1 and 7 in the morning when the law forbids use of electricity for this purpose. 93, 94. They use the bioluminescent bacterium Alivibrio fischeri, but the maximum lifetime of their product was 3 days. 92. In April 2020, plants were genetically engineered to glow more brightly using genes from the bioluminescent mushroom Neonathopanus nambi to convert caffeic acid into luciferin. 94, 95. Another possible application is to replace chemiluminescence with bioluminescent enzymes. A Canadian company, Lux Bio, is developing long-duration bioluminescent enzymes for this purpose. 96, 97. ATP bioluminescence. ATP bioluminescence is the process in which ATP is used to generate luminescence in an organism, in conjunction with other compounds such as luciferin. It proves to be a very good biosensor to test for the presence of living microbes in water. 98, 99. Different types of microbial populations are determined through different sets of ATP assays using other substrates and reagents. Ranilland-Gaussier-based cell viability assays use the substrate cholinterazine. 100. See also. Animal coloration. Biophoton. Life That Glows, 2016 full-length documentary. Notes. A. However, the name phosphorus, as used in the 17th century, did not necessarily mean the modern element. Any substance that glowed by itself could be given this name, meaning light bearer. 14. References. 1. Pless, Merrick, the 11th of November 2021. Lab snapshots by Merrick Pless. Microbiology: The biology on a different level. WeirdScience.eu. Retrieved the 2nd of July 2023. 2. Callaway, E. 2013. Glowing plants spark debate. Nature, 498, 15-16, the 4th of June 2013. 3. Smiles, Samuel, 1862. Lives of the Engineers. Volume 3. George and Robert Stevenson. London, John Murray. p. 107. ISBN 978-0-7153-42817. ISBN refers to the David and Charles reprint of 1968 with an introduction by L.T.C. Rolt. 4. Fries, Barbara, 2006. Cole, A Human History, RCKC and PG equals PA 51. Arrow, p. 51. ISBN 978-009-94788443. 5. Harvey cites this as Baker, J. 1743-1753. The Microscope Made Easy, an Employment for the Microscope. Stephen Haddock et al. 2010 list as more or less definite functions in marine organisms the following: defensive functions of startle, counterillumination, camouflage, misdirection, smoke screen, distractive body parts, burglar alarm, making predators easier for higher predators to see, and warning to deter settlers, offensive functions off lure, stun or confuse prey, illuminate prey, and mate attraction, recognition. It is much easier for researchers to detect that a species is able to produce light than to analyze the chemical mechanisms or to prove what function the light serves. 33. In some cases the function is unknown. A Swiss species in 3 families of earthworm, Oligocata, such as Diplocardia longa, where the coalomic fluid produces light when the animal moves. 48. The following functions are reasonably well established in the named organisms. Counterillumination, camouflage. Principle of counterillumination, camouflage in firefly squid, Wattisenia scintillans. When seen from below by a predator, the bioluminescence helps to match the squid's brightness and color to the sea surface above. In many animals of the deep sea, including several squid species, bacterial bioluminescence is used for camouflage by counterillumination, in which the animal matches the overhead environmental light as seen from below. 49. In these animals, photoreceptors control the illumination to match the brightness of the background. 49. These light organs are usually separate from the tissue containing the bioluminescent bacteria. However, in one species, Euprymna scolopes, the bacteria are an integral component of the animal's light organ. 50. Attraction. Storoteuthis cirtensis bioluminescent photophores. Bioluminescence is used in a variety of ways and for different purposes. The serrate octopod Storoteuthis cirtensis emits bioluminescence from its sucker-like structures. 51. These structures are believed to have evolved from what are more commonly known as octopus suckers. They do not have the same function. Storoteuthis. 50. Attraction. Storoteuthis cirtensis bioluminescent photophores. Bioluminescence is used in a variety of ways and for different purposes. The serrate octopod Storoteuthis cirtensis emits bioluminescence from its sucker-like structures. 51. These structures are believed to have evolved from what are more commonly known as octopus suckers. They do not have the same function as the normal suckers because they no longer have any handling or grappling ability due its evolution of photophores. The placement of the photophores are within the animal's oral reach, which leads researchers to suggest that it uses its bioluminescence to capture and lure prey. 52. Fireflies use light to attract mates. 2 systems are involved according to species. In one, females emit light from their abdomens to attract males. In the other, flying males emit signals to which the sometimes sedentary females respond. 48. 53. Click beetles emit an orange light from the abdomen when flying and a green light from the thorax when they are disturbed or moving about on the ground. The former is probably a sexual attractant but the latter may be defensive. 48. Larvae of the click beetle Pyrophorus nyctiphanus live in the surface layers of termite mounds in Brazil. They light up the mounds by emitting a bright greenish glow which attracts the flying insects on which they feed. 48. In the marine environment, use of luminescence for mate attraction is chiefly known among ostracods, small shrimp-like crustaceans, especially in the family Cyprididae. Pheromones may be used for long-distance communication, with bioluminescence used at close range to enable mates to home in. 33. A polychate worm. The Bermuda fireworm creates a brief display, a few nights after the full moon, when the female lights up to attract males. 54. Defense. Acanthophira purpurea has photophores along its body which it uses in defense against predators. The defense mechanisms for bioluminescent organisms can come in multiple forms: startling prey, counter-illumination,